Hi, and thanks for using the YSP DNA extraction kit. My name is Stephanie.
And I'm Ron.
And today we're going to just walk you through the protocol of the DNA extraction kit and suggest some ways that you as the adult putting on this demo might be able to engage the students in discussion.
So one way you can start is to ask the students to look around and ask them what physically do they notice are different between each other or same between each other. And you can write down their suggestions and their answers on a board just so they can see visually, what's different, like hair color, eye colors. Some kids are shorter, some kids are taller, or they're the same height. Just list those differences and similarities. And then after you've given them this opportunity to give this list, you can tell them these differences are basically due to differences in DNA. And then you can follow that up with asking the students, does anybody know what DNA is?
And you'll probably get a lot of random answers. Students really have heard about DNA from TV shows like CSI or any number of places. So the students will have ideas about what they think DNA is, and any answer is a good answer. The goal really is just to keep the students engaged and interested in what's happening here with the kit. And so after you engage them as to what is DNA, you really want to make sure that they understand that it's the DNA that is what makes me different from wrong. And what's really cool about the DNA is just a tiny fraction of it is different between he and I. Only 0.1% of our DNA is different, but it makes us so unique.
And because of that uniqueness, we can use that as a tool for crime investigation or to figure out family members and do all kinds of cool stuff with the differences in DNA.
Right. And so once you've described to the students what DNA is and why it's important for us, because it is the building block of life, you can ask them if they know where DNA is located. And they might not know actually where DNA is located, but again, you should try to squeeze out some answers from everybody and try to write them on the board to keep the students engaged. And in the end, you want to make sure that they understand that DNA is located in the nucleus of cells. And you can spend a little time trying to describe to them what is a cell.
So you can explain to them, cells are found all over your body. There's cells in your ears, there's cells in your eyes, and your hair, and that's why, for example, in crime investigations, all they need is some blood or some hair, and that contains the DNA, which contains the differences between one person and the other, and they can use that for forensics or crime investigation.
Right. And the truly amazing thing is that it just takes such a small amount of any one drop of blood or one piece of hair is good enough to get all of this information because of how many cells are contained within that drop of blood or within that strand of hair. And because the way DNA is passed on from cell to cell is by replication and division, every single one of those cells will have the exact same DNA in it. So it's essentially amplifying this really small amount of DNA so that we can look at it in a large scale from just one piece of hair because there's so many cells.
So to summarize, there are 2 points that are important to get across to the students. And the first point is that DNA is what makes us different or same.
And then the second point is that the DNA is found in cells and that every cell has the exact same amount of DNA within one organism. Okay, so the first thing you want to do whenever you're starting a science experiment is to make sure that you have every single thing that you need. And so in this experiment, what the students are going to do is isolate DNA from fruit like strawberries or from peaches. So let's make sure that we have everything we need.
Salt, some stirring sticks, at least 2 plastic cups, some alcohol that's cold.
You'll also need water, shampoo, and then the fruit you're extracting your DNA from, so peaches and strawberries. In addition, have gloves so you don't get too dirty. And you'll also need pieces of cheesecloth cut so that they can be placed over the cups. Rinse the peaches and the strawberries, and you can do this before the students arrive, especially if you have a lot of students. You might want to just rinse the entire bag of strawberries at one time. But in this case, we're just going to show you a little demo, and all I'm going to do is rinse exactly what we need for one experiment. And so what you want to do is just grab, whatever, a small handful of strawberries about like this, and go ahead and rinse all the sugar from the packaging off of the strawberries. Just throw them in a cup. And, just rinse them a couple of times. Okay, good enough. And then you're going to want to do the same thing with your canned peaches. And so you'll just take 2 or 3 of these little quarters of peaches. And you can put them in a cup to rinse them. And again, we'll just give them a nice quick little rinse. Okay. And that's the first step. So all you need is about 1/4 to 1/2 cup of the produce.
Now that you have your produce ready and washed and you have all your ingredients, always remember throughout the experiment to always wear gloves. So step 1, the goal of step 1 is to basically break open the fruit and break open the cell by a smashing process, physically smashing the fruit with your fingers. And we're going to demonstrate how to do that.
So kids might really like to smush the fruit up, and it can be a little messy, so you might want to have paper towels on hand, but it doesn't have to be a complete liquid. Small chunks will be okay. So after a couple of minutes of smushing, the strawberries look about this, and peaches look good, so we're ready to move on. Okay, so now that the fruit is all smushed up, the next step is to add an equal part of water, and it really doesn't have to be exact. So I'm just gonna eyeball what I think is equal. That looks good to me. So Wrong, go ahead and do the same with his peaches. Okay. And then after adding the water, we're going to proceed with a lysis step where we'll add a detergent, in this case, shampoo, to the mixture. So we just need a couple squirts of shampoo, and again, doesn't have to be exact. Okay. Great. And then after that, we add just a small, like a teaspoon of salt, which I'll just approximate in my hand. That looks good to me. And then very gently, we can just use a finger, or Ron can use a popsicle stick, and we just gently mix these together. And you don't want to be too vigorous at this step because we want long, gooey strands of DNA at the end. Okay. Now in the final step, you're going to actually precipitate the DNA out of solution, and you'll do this by using ice cold rubbing alcohol. And so what I'm going to do is just approximate an equal volume of alcohol to add to my soupy mixture here, and use a popsicle stick and look at all this goopy DNA. That's a lot of DNA. And Ron can go ahead and he can do the exact same thing with the peaches. And so you can see a little bit of DNA here with Ron's peaches. And what might be fun to do with the students is ask them to make some observations throughout the experiment and as to why I have more DNA in my strawberry than Ron sees with the peaches. And some possible explanations for this that make a lot of sense are that I used more strawberries than Ron used peaches. But other equally good explanations are that strawberries are full of seeds and little cells on top of cells on top of cells. And so there might just be more cells in a strawberry than there are in an equal volume of peaches. So it's a little bit messy, but in the end, the goals of this are to show students that you can use household items, everyday items that you can buy in a grocery store to do things that scientists do in labs or from crime scenes all the time, and that it's easy to do and sort of fun.
Now that you've completed the experiment, be sure to remember to clean up after yourself. And everything that we've worked with here, you can just basically just dump down the drain. And whatever is left over, you can just go in the trash can.
And then the last thing to do after you do any experiment is to sit down and spend some time thinking about your results and the methods you used and trying to figure out why things worked the way they did. And so we've included with the packets a series of questions that you can provide the students to get them thinking about what we talked about today and what DNA is, where it's found, and why it's important. In addition, we also ask that you please fill out our surveys and get them back to us, because our goal is to make these kits as useful for you as possible. So any feedback you have, maybe this video wasn't helpful, maybe it was. So just let us know what you thought, and any improvements you could suggest are greatly appreciated.
Thanks for watching our video, and hopefully we drove across the point of the importance of DNA and cells, and basically give you a helpful overview of how the experiment may go.
Thanks.